Dr. Paushali Pal explaining you the modern technologies involved in microbiology. So I would request you to watch my video till the end. Thank you so much.
Okay, so after the introductory video, let's go on with the slides. Topics for discussion include electrospray ionization mass spectrometry, Malditoff MS, ion spray method, capillary zone electrophoresis, NAT using biosensors. Then new technologies for infection control will be one point of discussion that include electronic hand hygiene monitoring systems, antimicrobial textiles, and ultraviolet C devices. So let's get on with electrospray ionization mass spectrometry. As you can see, this is the instrument. This is the instrument I'm pointing out. And this, you can see a nozzle kind of thing is protruding out through which the sample comes out in the form of ions, and the electric field that we are talking about is inbuilt in this machine or in this kind of equipment. So what happens? The sample molecules are sprayed in the electric field that is present here, inbuilt over here, to produce ions separated by mass charge ratio. So the sample comes out through this nozzle in the form of ions, then it enters the electric field, and then it enters to the analyzation zone, wherein on the basis of mass and charge, the ions are detected. Analyzer then detects the ions based on mass charge ratio. So now, this is the sample along with the chemicals generating either acidic pH, in which we find positive ions are cations, or alkaline pH, in which we find negative ions or anions. Generally, the cations are more useful for any kind of sample analysis. So we go about with generating acidic pH to form cations. Suppose this is the sample. As you can see, this is the nozzle kind of thing through which the sample is carried over in this vacuumized zone, and then the sample with its watery and chemical components get vaporized to leave only the ions. How and why it gets vaporized is because the sample and the charge is all the same. Due to repulsion of these two charges of the sample and the sample watery part of it, it gets vaporized, like the solution part of it gets vaporized, and the ions remain, which then go on to the detector for analysis. See this, the ions, basically the very tiny ions, they go on to the analyzer part, and the analyzer analyzes it on the basis of mass and charge ratio. Always remember, smaller the ions, greater will be its intensity of charge or charge intensity, and so it will fly more faster onto the detector rather than the bigger ions. I mean to say, smaller the ions, lesser the time of flight onto the detector. Likewise, smaller ions go first on the detector, followed by the larger ions. On the basis of that, the ions are separated, peaks are generated and analyzed. Ions are analyzed. See here, more clarity on it. The bigger ions and the smaller ions. Obviously, as I told you, the smaller ions will overtake the bigger ions and hit the detector faster, creating peaks closer to the zero origin rather than the bigger ions. Next, we go on to MALDI-TOF-MS, that is matrix assisted laser desorption ionization time of flight. This is the machine. As you can see, MALDI ion source, one ion source has to be there, time of flight of the ions has to be there, and then the detector. So what is it basically? Let's go on. The principle involves necessarily the creation of the ions with the help of laser. L is for laser. Matrix assisted laser desorption ionization. Laser beam is involved. So as laser beam excites the sample present in the matrix, the ions are generated, and based on mass charge ratio again, smaller ions hit the detector faster rather than the bigger ions. The same thing. Here also the same thing, only here the laser is involved. Remember this. In MALDI-TOF-MS, L, laser is involved. As you can see here, this is the origin. Closer to the origin, the pink peak, that is from this pink colored ion, hits the detector faster. So the closer to the zero origin, the pink colored peak is generated, while the green color ion is far from the origin. And then the time of flight is measured. Accordingly, the peaks are generated. Obviously, the more tiny the ion will be, the lesser time it will take for it to hit the detector. Whereas the larger ions take more time to hit the detector. This is the principle, fundamental thing behind it. So how does this laser create the ions? This should be your question. Laser beam is incident on the matrix, that is finely grained material, along with the sample. These blue ones that you can see are the samples. As the laser beam hits the sample present in the matrix, that is within the crystal, the ions are generated. And these ions then go on to the mass analyzer based on again mass charge ratio and ion intensity, charge intensity. The ions are detected, that is peaks are formed. See here, the sample molecules are indicated with red, surrounded by matrix molecules, and then the laser is incident on this matrix along with the sample. Then it gets protonized. Once the laser is incident on the matrix, the sample gets protonized, meaning to say electron is lost from the H neutral H atom, give it a proton H plus along with M. Okay, so MH plus is formed. Fine. So protonization happens for the sample molecules. And then what happens, you know very well. The ions are generated, and the mass charge ratio, on the basis of mass charge ratio and charge intensity, it hits the detector at different speed, and peaks are generated at different locations. So now let's go on to ion trap method. This ion trap method is a combination of electric field and magnetic field along with radio frequency, and it captures the charged particles or ions within the hyperbolic rings that I'll show you. And then what happens, we will get more clarity. This is the machine. This is the machine equipment. Ion trap method, as the name suggests, ion trapped. Ion is trapped. So ions are injected from the source. You can see differently colored ions are injected from the source onto this hyperbolic rings on all sides. And when they are inside it, what happens? Apart from the precursor ion, we make the other ions out of it and make them incident on the detector. Okay, only the precursor ion or the ion in which we are interested is made to remain on the center, and the other ions are out onto the detector. Okay, they're also on the basis of mass charge ratio and charge intensity. It hits the detector at different speed. Then what happens? The charge or the ion in which we are interested, that is the precursor ion, is made to bombard onto the walls. Just like imagine a carom board, a striker is being struck on the boundary of the carom, and imagine the striker to be splitted into parts. Just like here, the striker, that is the ion of interest, is splitted into parts on hitting the boundaries of the carom, just like the boundaries or walls of the hyperbolic ring over here, and they are being dispersed into small pieces. These small pieces of ions, fragmented ions, are made incident onto the detector based on mass charge ratio and charge intensity. It hits the detector at different speed, creating peaks closer to the origin or farther from the origin according to its size and charge intensity. Obviously, you can see the smaller ion hits the detector faster rather than the bigger ion part. So that's about the ion trap technology. Then let's move on. Hybridization probe. Hybridization probe, it actually wants this portion of the DNA is cut from a big DNA, and suppose we want to visualize this DNA. So for that we will do what? We will add a probe. That is an oligonucleotide probe, and to that probe we would like to add primary antibody, then secondary antibody labeled with enzymes such as horseradish peroxidase for visualization. Remember, hybridization probe, whenever we use the probe, that means it is used for visualization. For visualization, we need the probe in actual, we need the primary antibody, we need the secondary antibody along with enzyme, horseradish peroxidase. And then we fix all this onto the probe, and a particular segment of DNA is visualized. Now for visualization purpose, using probe, we need to hybridize with the probe also. This is the probe sequence. It's just like the highlighter that I'm using here. It's used for visualization, having a sequence A, T, C, A, G, A, T, C, A, G, T, A, C, C, A. As you can see, this is the probe sequence. Does it match with the first one? No. A with C doesn't match. T with A matches. C with T doesn't match because you know already adenine makes bond with thymine and guanine makes bonds with cytosine, AT and GC. So it is the second sequence which matches exactly, and the probe sequence hybridizes or makes bond with this second sequence that I have shown here, with this second sequence, and hence this part of the DNA is visualized since the probe sticks to the target in this way. Target means that particular sequence from the whole DNA. Okay, so this is hybridization probe. This is how the hybridization probe is used for diagnostic purpose also. Let's go on to the capillary zone electrophoresis. This capillary zone electrophoresis is just like ordinary electrophoresis, except for the fact that it uses fused silica capillary. It has several advantages like high separation efficiency, rapid analysis, minimal sample consumption, compatibility with various detection methods like UV MS, ultraviolet mass spectrometry, fluorescence MS, high resolution separation. Now let's talk more about it. See, capillary zone electrophoresis, as I said, uses fused gel. So you can see the fused gel is running already through this, and through this fused gel we have the anode inlet and the cathode outlet within the machinery. Anode inlet, cathode outlet. Anode, as the name suggests, it attracts the negative ions of the sample, while the cathode outlet attracts the neutral and the positive ions of the sample. Okay, so the positive ions and the neutral ions get on to the attracted to the cathode outlet, generate the peaks, and the sample is detected. See, these are the three regions: injection end, that is this end, injection end, separation end, this part, separation end, and detection point, this detector, detection point. Now let's have more clarity. Electrolyte buffer is there, capillary inlet, then we have a separation region in which the negative ions go towards anode inlet, as we have seen in the previous slide, and the positive ions along with the neutral ions go towards the detector. Towards the detector, the positive ions hits the detector, creating peaks. It should be closer to the origin or far away from the origin based on again mass charge ratio and charge intensity. It creates peaks at different locations, and this is how the three portions of the capillary zone electrophoresis functions to actually detect the sample. High voltage is applied over here. This is how it works. Next, go on to capillary isoelectric focusing, that is for separating the monoclonal antibodies. This especially is used for diagnostic purposes. So what happens in this isoelectric focusing, meaning the pH at which the charge becomes entirely zero. Suppose this protein A and protein B starts from an initial point. Okay, starts from an initial point. When reaching pH near about 4.5, protein A, the charge of protein A becomes zero. That means this is the isoelectric point of protein A. Another green one, as you can see beside protein B, that also travels in the same way in another lane beside protein A, and its isoelectric point is near about 8.5. So what happens here? At 8.5 pH, its charge becomes zero, and then it stops here. So So, on the basis of charge becoming zero, that is, on the basis of isoelectric focusing, the proteins are separated onto the gel medium. This is what is electrophoresis based on isoelectric focusing or capillary isoelectric focusing. It is used to detect the monoclonal antibodies and thus used for diagnostic purposes. Okay, I hope this is clear. Next, we go on to NAT using biosensors. The full name is paper-based nucleic acid amplification test. Here what happens, just we had in the time of corona, RT-PCR is used for amplifying the sample RNA. Amplifying the sample RNA. After the sample RNA is amplified, the test is actually done. So capture probes are used. The capture probes will capture only a particular RNA from the virus. As you can see, this green one is captured by the RNA probe, that is the capture probe. This green one is captured, and then we wash or rinse it so that the other uncaptured other uncaptured samples go off. Then the captured probe, that is the sample which is captured by the capture probe, is visualized by the H2O2 has radish peroxidase enzyme, primary antibody, secondary antibody, and then we find whether the particular virus is present in the sample or not. Okay, so first what happens here is amplification of the RNA is done. RNA sample is made incident onto the capture probe. Capture probe captures the sample, then visualizes the sample. If that particular RNA is present within the capture probe, means that RNA is present in the sample also and the patient is rightly diagnosed. Now coming on to new technologies for infection control. Here we use electronic hand hygiene monitoring systems. It has two parts, trigger sensor and maintain sensor. Trigger sensor is fitted onto the sink where a person may wash his hands for 20 seconds and then it turns green. Initially it's blue, then it turns green. Then the maintain sensor, what it does, the maintain sensor is fitted onto the pocket of the person washing the hand, and as the person touches the washed hand onto his pocket, the blue signal turns into green. That's the work of electronic hand hygiene monitoring system, ensuring that your hand is clean enough. Next, we go on to antimicrobial textiles. These days, we have textiles which are treated ones by chemicals such as zinc, pyrithione, silver, silane quaternary ammonium compounds, silane quartz, polyhexamethylene, biguanide, PHMB. All these chemicals do one thing, that is they make the textile antibacterial, antifungal and antiviral, antimicrobial in gist. So let's go on. The first layer, as you can see, stops the bigger viruses more than 1 micron. The second layer stops the lesser dimension viruses and microbes lesser than 1 micron. And finally, you have the third layer through which nothing passes. I mean, no microbes can pass. And you have the fresh air onto your body. So these layers are made in such a way, or the textiles are made in such a way so that the microbes are unable to pass through the layers. Then this is the picturization part of it. As you can see, lotus microbe under microscope, lotus microbe filter, it filters the microbes. Ultraviolet C devices, the last topic, ultraviolet C devices. These are the fitted onto the rooms. Ultraviolet C devices, as we know, it kills the microbes based on DNA. It damages the DNA of the microbes. The DNAs are unable to replicate. That is, the microbes become dead. How the DNAs are damaged? DNAs of the microbes, how they are damaged is, this is the DNA, and what it does is, suppose the red color ones are the thymine ones, thymine, it clubs the two red color strands, that is thymine dimers are formed. As a result of that, this particular DNA is unable to replicate and the microbe falls dead. So thank you. Thank you for your patience. Meet you again in the next video. Thank you so much.